The technology I'm going to present today is the name of the application itself. A method of diagnosing infectious respiratory diseases through cell vesicles, that is, extracellular vesicle. Among them, I would like to show you the data applied by this technology from tuberculosis today. First of all, tuberculosis will still be a major infectious disease that remains a global public health problem. According to a 2016 survey, it still shows the 10th cause of death, especially in Korea, and although the prevalence has decreased significantly, it is still the #1 infectious disease among OECD countries. But in the case of tuberculosis, the rapid administration of anti-tuberculosis drugs, it will be the key to treatment, and in fact, various test methods are used to the routine process diagnose quickly. There are several methods here, which are usually used in the laboratory. First of all, acid-fast bacilli smear is used, and there is a gold standard culture method. And there are used as a screening tool such as tuberculin skin tests and IFN gamma-releasing FCETC, and molecular tests are used as another tool, mainly with the approval of the Ministry of Food and Drug Safety as in vitro diagnostic medical devices, usually PCR-based methods, especially real-time PCR techniques used. It is shown to summarize the advantages and disadvantages of each method currently used in the laboratory. First of all, AFB smear is an easy way for us to approach, but the biggest disadvantage is that the test sensitivity is not that high. Second, gold standards such as culture, which may be a variety of culture methods and common weaknesses, but because culture is too laborious and takes too long to determine the treatment. Although it has highest sensitivity and specificities, so as a complement to this, the nucleic acid amplification molecule, so to speak, molecular genetics methods are actually used a lot of routines. A wide variety of vendors have launched licensed products and are using different vendors in the laboratory. And it shows various sensitivity depending on the product or patient's condition. In particular, in case AFB smear is positive, the molecular genetic test methods are highly sensitive, and in fact, AFB smear itself is the first positive state, which is also used as a complementary tool to further increase PPV. It can be tested quickly, and while the specificity and positive predictive value are high, but it still has a little of a variety of sensitivity. And PCR itself can be seen as a disadvantage, and it has the disadvantage of the test results affection if there is a PCR inhibitor or contamination. Nucleic acid amplification, NAA tests mainly used in the laboratory as real-time PCR method. The biggest advantage is to be used clinically by detect the test results faster than culture. While population in AFB smear is negative, the positive rate is likely about 50% to 80%. It is one of the most used tests because results can be obtained quickly. And if you look at the last line, the nucleic acid amplification tests have the advantage of increasing the detection rate by multiple and various sample use. Because for the various in vitro diagnostic medical device products used in tuberculosis, most of the analysis methods themselves, that is, downstream analysis, are clearly defined as in vitro diagnostic medical devices (SOP) and an advanced step at each laboratory. There may be cases to choose the best way how to isolate and extract those. Another thing is that we have to think a lot about the free underrated valuable value before the analytical step, which has a great influence on the final test results. So the sampling can affect the test results. Therefore, it is recommended that we obtain not only ordinary spurt samples, but also various samples that can be obtained from patients, at least more than one. So, the first thing we came up with is to separate the exosomes from the patient's sample. Exosomes are a kind of extracellular vesicle that originates from various cells in the body, usually nanoparticle as small as 30 to 100 nanometers. As you can see here, the various membrane proteins in the membrane and things that reflect the origin of a wide variety of cells are included in the components. So it will be a field that has been used a lot, especially in the cancer diagnostic area recently. As you can see in the center, it also contains hexane, DNA, or RNA, so we decided to use this ingredient to prepare samples with these in mind. By this, particles originating from cancer cells can be used in antigen-presenting cells, and after that, they can be included in various cell origins and the cells in the body, as well as the entering the bacteria from outside. These are the parts as samples because these are included in exosomes ingredients. In particular, experiments have shown that exosomes are actually isolated from the MTB-infected animal model, mice model, and that TB inside exosomes has proteins. So we tried to isolate exosomes in the process of patient samples handling in real routines based on these results. Another thing was to separate the exosomes and then use the ones used downstream called digital PCR method use. The second is shown the quantitative PCR, or real-time PCR, which is the most commonly used in the routine process. And digital PCR has a way to endpoint detection. If you look at it as a singularity compared to quantitative PCR, next time absolute quantification is possible. Therefore, we wanted to detect the tuberculosis through exosomes and digital PCR combination methods with the advantage of being able to improve sensitivity, because it has the advantage of being able to perform quantitative analysis without any standard cover. Basically, the principle of digital PCR may vary depending on the platform, but the most basic principle is partitioning. So the basic principle itself is to use self-acting chemistry and microplates. In fact, we can consider PCR as a PCR mixture in a small room with several rooms and tens of thousands of rooms, each designed to emulate single molecular targets. When the amplification target exists, it becomes the target we want. The fluorescent dyes that we put on in advance emit the fluorescence to be able to detect. Now, why partitioning can help increase sensitivity. And assuming on a single tube that amplifies a bulk sample amplification with multiple targets, and especially variant that we want to see as Warren status, the percentage is very low. But if we divide this into small rooms, the variant target we want from each one will be in rec effects, which will ultimately increase the sensitivity, because it has the effect of increasing the concentration, and that is a major principle. But intuitively we can calculate that if a single molecule is included in one of these droplets, we can simply calculate that some of the 20,000 droplets were created. But in reality, due to 2 or 3 in droplets, actual absolute quantification method is included in the droplets. So we can actually use statistical distribution model Poisson statistics. So it can be seen intuitively. And the blue dots and the black dots correspond to droplet, respectively, and we can intuitively check the two-dimensional graph with 1 existence and 0 non-existence. When it comes to using ddPCR for pathogen detection, it can have advantages, especially because it is possible to use it in various places, such as extracurricular TB. Especially when the number of copies is very low, the increased test method using is important. So it can be used to various area, and recently we use the usefulness such as the detection of cfDNA in the blood. Therefore, in the area of infectious diseases, it will be a way to use it because it is helpful to diagnose or to improve sensitivity. If we show you the results, we have targeted 190 patients who have requested specifications. Here, 44 patients are finally confirmed patients with pulmonary. If you look at the types of respirational specifications, there are various types. In addition to sputum, bronchial washing, and BAL were all available, and because exosomes themselves exist, very ubiquitous. We have the advantage of being able to isolate not only blood, but also various body flows, so it can be applied regardless of sample type. So, we have used ddPCR using the target that is most commonly used to detect tuberculosis. But since the process of exosome separation is actually important, we need to know the physical properties of the extracellular vesicle. If you look at the bottom, the extracellular vesicle can actually exist within a variety of sizes. So there can be a wide spectrum ranging from about 30 to 5,000 nanometers, and there can be spectra in which things like live proteins that exist in the body overlap very similarly, and viruses can exist similarly, and there are many attempts to isolate them by separating them into density on the y-axis and size based on the x-axis. And in fact, there is no standard method to isolate them, so a wide variety of researchers are trying them. This is because, as I told you previously, the spectrum that has to be differentiated from many things overlaps. And in fact, recently, when used for treatment purposes such as drop delivery in the cancer, it has to be high purity with isolation. So, we are trying many things by using various charges, such as size, or using a variety transfill to increase yield, while increasing purity. We used a commercial kit for this experiment, which is actually not known because intellectual property rights are protected, and then the technical scope includes the composition of reagents that can be used for treatment. So we can see the same test performance in the same sample later. This is the analysis result. In confirmed patients, you can see how to extract total DNA, a common isolation method, and ddPCR, which are AFB smear and qPCR commonly used, and how to extract exosomes, and methods by dividing total and exosomes. Since the specificity is all high, if you pay attention to sensitivity, AFB smear is 34% and qPCR is 47% when total DNA is selected. And you can see that it improves to 54% only by changing it to exosomes. The ddPCR was 61% when total DNA was used and 75% when exosomes was used, showing a 27% increase in qPCR, which is commonly used, compared to total DNA. And culture results show the highest test performance at 88%. This is because the number of droplets can be checked in a culture-positive sample, especially in ddPCR, so we collected the positive droplets with less than 100 droplets. And if you look here, it can be now shown more positive signals in the same ddPCR and qPCR. Even if the same ddPCR is applied, it can be seen that the positive rate drops to 53% when total DNA is used, and the detection rate drops by half in qPCR as well. So, we think that this particularly low load means that hexane is more stable than it has in cellular plasma. In summary, we thought that exosomes could be used as a useful target for detecting nucleic acid derived from pathogen. Today, I showed you the results of applying digital PCR, and I think we can apply pre-analytic things to various platforms that we are already using for diagnosis in various sample prep stages. And in case of the combination of the DNA of ddPCR and exosomes, it would be that the detection rate could be increased and the accuracy was also high in detecting tuberculosis. In the low concentration sample, different detection rates for each sample type appear to suggest that they exist statically in exosomes. So the respiratory specimens are actually more similar in clinical symptoms, so the current platform is being developed toward multiplexing. Samples are the same, but the development of a platform that detects various things simultaneously is the direction of progress. So we plan to expand and evaluate the test performance for different pathogens, such as viruses and bacteria, using exosomes. That's all.